Hello everyone. So today we're going to be talking about um, our first unit, which is pharmaceutical quality and current good manufacturing practice. So you'll see this bar down here. You have your home screen, which will take you back to your first slide. You'll have Unit 1 learning objectives. For the first unit, I have taken information from the presentation in the book, and I have using those two resources to answer the unit learning objectives as if the unit learning objectives were questions. And that is a good practice throughout this course to be able to take some of this information, which is a lot of information, from multiple sources and synthesize it into one place to help you study. I have done that for you for the first set of unit objectives. Moving forward from Unit 2 on, if you want to do that, you're going to need to do it on your own. But I will show you what that looks like. So we're going to go into the Dropbox link, and it shows you the Unit 1 learning objectives with answers. Okay, and you can go through that. Once I'm done making this video and I post it to YouTube, I will be hyperlinking this to YouTube, so you'll have a lecture video, so that way if you're sick or you miss class for some reason, you can watch the lecture video. That being said, I've said it before, I will say it 1,000 times, do not come to class sick. I do not want to be sick. This is why I make all these resources, so that way you don't come to class sick. Do not come to class sick. Don't do it. There's flashcards. For this unit, I've broken them down into smaller sets. Moving forward, it's just going to be one big set. This is going to take you to Quizlet. From Unit 2 onward, if you want to copy that Quizlet and make that Quizlet into smaller sets, you can. So this is what it's going to look like for the first set. It goes through those learning objectives that I just showed you. It goes through learning objectives 1 through 10, 11 through 20, 21 through 30, and so on until the end of the objectives. Okay. Lab. Here you're going to have your lab documents. If it's not a wet lab, then you can go through it at home. If you do miss a wet lab, it's going to be a little bit difficult for you to make it up. But if it's a dry lab, meaning that you don't have to have like equipment or chemicals or anything, then feel free to do the lab at your leisure if you miss class. Lecture. When you hit this lecture tab, you're going to need to hit it one more time to get to the sections. Now these sections are hyperlinked, so let's say I missed section 4. I click on section 4, it takes me automatically to section 4. Okay. Let me get to the end of section 4, so just bear with me for a minute, and I'll show you the review. So for Unit 1, I put a small review at the end of each section, again showing you how to break down some of this information into smaller bits to be able to process it better. Move from Unit 2 on, I'm going to have one review section at the end. Okay, so if you want to break it down into smaller parts, after Unit 1, you're going to need to do that on your own. But here's a review slide, so I'm going to give you time to go through the learning objectives. Again, here's the hyperlink for it, for 21 through 30. Oops, let me go back. This set 3 goes with section 4, and there's a game with it, and so the order I suggest you go in is to do your learning objectives, synthesize them into notes, master the flashcards, and then go to the games. The games are a bit harder and they're timed, and so to not get so frustrated with the game, I suggest learning objectives or slows, flashcards, then game. But whatever works for you, whatever floats your boat. For assignments, you're mainly going to get graded on quizzes. And so it says assignments. This is supplementary material, so that way you can have good conversations and informed conversations when you walk into the pharmaceutical industry after taking this class. So let's jump right into the lecture section. These are things we're going to be doing in class. You can go through it on your own time if you're doing it at home. So the mission statement of the Food and Drug Administration. The Food and Drug Administration is responsible for protecting the public health by ensuring the safety, efficacy, and security of human and veterinary drugs, biological products, and medical devices, and by ensuring our nation's food supply, cosmetics, and products that emit radiation. The authority of the FDA comes from the USC, or United States Code. The United States Code is approved by Congress and is the ultimate authority that gives FDA its legal authority. The specific law that allows the FDA to take legal authority is the Food, Drug, and Cosmetic Act, and the goal of the Food and Drug Cosmetic Act is to take legal action against adulterated or misbranded medicines. Adulterated medicines means that something has happened to the purity, strength, potency, or sterility of the medication itself. Misbranded means that the medication, although it could be pure, potent, and all that good jazz, it means it has been misrepresented. So the responsibilities of the FDA include reviewing or rejecting applications for new drugs, overseeing the manufacturing of said drugs, reviewing and approving or rejecting marketing materials to make sure that the material is branded correctly. So the marketing materials are a part of that labeling component that we just talked about. The FDA performs its own independent research to make sure that the data presented by pharmaceutical companies for new drug applications is scientifically sound. The research is not done for the pharmaceutical company. The pharmaceutical company has to do their own research. The FDA comes behind and just validates their research. There are 3 types of inspections, and so you're going to have your general inspections that happen every 2 to 5 years, your run-of-the-mill inspections just to make sure everything's running smoothly. You're going to have pre-approval inspections, which are inspections that the FDA goes through when there's a new drug that's looking to be approved. So the manufacturing of that new drug, the FDA wants to make sure that that is going smoothly. If there is a complaint or an issue with a medication, those could be for-cause audits. It means that there was an issue, and so the inspection is not a run-of-the-mill inspection or an inspection to approve a new drug. These are audits that have a cause. The Code of Federal Regulations is part of the Food, Drug, and Cosmetic Act, and it is comprised of food and drug regulations, and it sets the expectations that biomanufacturers need to meet to fulfill the requirements of the Food, Drug, and Cosmetic Act. CGMP stands for Current Good Manufacturing Practices. Good manufacturing practices is the minimum standards set by the Food and Drug Administration. Should you choose to work in pharma, you will hear GMP every single work day for the rest of your time working at a pharmaceutical company. Every single day. So you need to know what it means. 21 CFR sets the guidelines for every aspect from labeling to release of product for distribution. Labeling, everything is tightly regulated, but we will talk about labeling a lot. Labeling is very heavily controlled because it's part of good documentation and data integrity. A drug is considered safe if its benefits outweigh the risks. A drug is considered effective if in clinical trials a drug is shown to have to be more beneficial than a placebo. So to comply with FDA requirements, you need to review your documentation and records to make sure they're complete and accurate. Making sure that samples are properly labeled and handled within laboratory practices. Observing how process technicians do their jobs. And seeing how clean the environment is. So the current input in the Good Manufacturing Practice acronym is indicative of the fact that processes keep improving. And so because processes keep improving, you need to stay current with the best manufacturing practices. And so that's what that C emphasizes. No person can work in the biopharmaceutical industry without understanding these rules. People in the industry have a CGMP mindset. That means they learn to think about what happens, how their actions affect patient impact. Quality has two facets: safety, which we already talked about, and efficacy, which we already talked about as well. These are some of the downsides to poor quality. Regulatory agencies such as the FDA notify companies when they find operational deficiencies, such as when they're going through those different types of inspections that we talked about earlier. Depending on the severity of the problem, the agencies such as the FDA may give a time limit to correct the problem, or maybe even block products from being marketed. So the quality unit within a pharmaceutical company is considered separate from other departments. That's because the quality unit has to focus on quality, so someone who is a process technician under quality doesn't have to report to someone in marketing who has a different priority outside of quality. Okay, so quality assurance oversees overall site quality, quality control, test raw materials. You have different quality systems that maintains capturing deviations, corrective actions, and so forth. Training is important because the biggest cause of deviations is inadequate training. The biggest cause of deviations is inadequate training. The quality assurance staff reviews batch records, performs calculations, reviews documentation, and the quality unit as a whole is responsible for managing and completing a number of documents. They're also responsible for performing internal audits. You want to perform internal audits so that way if there are any issues, you can catch those issues before the FDA does their own audit or inspection. So rules for quality: say what you do, have a standard written procedures, do what you say, follow those procedures, prove it, keep records, and improve it. And this is where you would take the time in the class to review. So documentation. If it is not documented, then it didn't happen. So story time. When I was a really broke community college student, I went to used car lots because I only had $1,000 and I needed a car in order to keep being able to go to class. I went to one particular car lot. The car dealer told me, well, this car is kind of expensive because I just had a new engine put in it, but I lost the paperwork, right? Should I believe him? No, I should not, because if it isn't documented, then it did not happen, and the FDA will tell you the same thing. If it is not documented, then it did not happen. You cannot prove that it happened. You have not kept a record that it happened. Good documentation practices applies to all the list of these things on this slide. Standard operating procedures, labels, batch records, laboratory data. All these things require good documentation practice. Alcoa, you will need to know Alcoa for any job interview in pharma. You will need to know Alcoa for any job interview. Attributable, who, when. So who wrote it and when did they write it? It needs to be legible. If you don't have legible handwriting, then practice until you do, period. Contemporaneous means that it is documented as it is happening. I don't go in tomorrow and record what I did today. Original means it's on the original paper, original batch record, etc. Accurate means that it's correct. More on good documentation practice. Your documentation needs to be complete, meaning that all information is present. It needs to be consistent, meaning that all information is recorded in the order that it was generated. Enduring, it needs to be an appropriate medium, such as paper or electronic, so that way it can be accessed later. And by being able to access later, it needs to be available. All these things is why it's important to have a record retention policy, and every pharmaceutical company will have its own record retention policy. Data integrity refers to the completeness, consistency, and accuracy of data over its life cycle. So all of these elements contribute to data integrity. So the underlying premise is that in order to show that the drug is safe, that it is potent and pure and all that stuff, you need to have good data integrity, because the data is what's going to prove that. If you cannot prove that through data integrity, then it didn't happen. General rules for completing documentation. You need to use permanent, also known as indelible. A lot of people in the industry use the term indelible, which just means permanent ink. You need to be able to write legibly. Entrances should be complete. Data should be correct. You need to date your signatures. Every pharmaceutical company has its own procedure for dating. A lot of times time will be in military time. Do not use ditto marks to indicate that the entry is the same as line above. Write out each entry. Now, this is very important. Do not, if you write down the data wrong, do not try to change the data. You need to correct the data. And what is the difference? Changing would be turning a 3 into an 8 or a C into a D. That is changing. It looks like you are trying to change the data, and that can take away from your data integrity. Now, if you appropriately correct the data by putting a line through it, writing what's supposed to be there, initialing, dating, putting a footnote, and at the bottom of that footnote, you also initial and date and state why the correction had to be made, such as you wrote the wrong year or something like that. Do not use correction fluid, erasers, or any other means to obscure data for the same reason. It can take away from data integrity. It can look like you're trying to create false data. Never backdate an entry. Some things, such as batch records, will require a second person to verify that a certain step in the operating procedure happened. If you are a verifier, you need to actually be verifying. Do not let someone come up to you and say, hey, can you just sign this for me? I did it. Your eyes need to be on it, making sure it's being verified, because if something later on down the line happens and they trace it back to that particular step in the SOP and you verified it, then it's you on the line as well as that other person. Okay. If you have a section that's not needed, do not leave it blank. When you leave it blank, it looks like you could be trying to leave out important data, which is why you want to put NA for not applicable instead of just leaving it blank. When you leave it blank, that again takes away from that data integrity, because it looks like you might be trying to intentionally leave out data. Okay. This is a short video here about correcting documents. I'm not going to show it here. You can look at it. But it does show a good example. And I just walked through these steps already. But this video does do a good example of showing that. And take some time to review. Section 4, Facilities, Equipment, and Material Controls. And so the facilities are going to have 3 things coming in and 3 things going out. You're going to have raw materials coming in, people ready to work to come in, and utilities such as, water, HVAC, that sort of thing coming in. Going out, you're going to have finished products, personnel who leave work, as well as waste and scrap. So facilities need to be of suitable size and construction for what's going on in those facilities. There needs to be adequate space for the orderly placement of equipment. You're not trying to squeeze things together. You're not trying to make things fit. When you don't have quite enough room, facilities are built to do what they're supposed to do effectively and safely. The flow of personnel should be unidirectional if possible, and everything shall be of adequate size to prevent contaminations or mix-ups. And also, you need to have separate or defined areas. One particular company I worked at, when you went into the production area, you had one production area for solution prep. Solution prep was done there. There was another production area for the filling of vials. You would never switch those rooms, so you're not going to take the stuff from solution prep and put it in the room where you're filling vials. It doesn't make sense. And there's a risk of contamination to occur. When you're talking about cell culture, that was a whole different space. So you're not going to take and have a room with cell culture and then have a room in the same room have where you're filling vials. Okay, everything has to have a designated purpose and space. And these are the examples of some designations for facility rooms. So for aseptic procedures, you have what's called clean rooms, and so you have to go through a lot to be able to get into those clean rooms. And so A would be the cleanest, B, C, and D. When I worked in a C clean room, what I had to do was starting from the outside, so coming in to the locker room in my street clothes, I would have to change into a set of scrubs, and then I would have to step over this particular line. On one side, I would have my dirty shoe. On the other side, I'd have to put on a clean shoe. And I couldn't put on the clean shoe until I stepped across that line, but I couldn't step across that line in that dirty shoe. And then I had to put on gloves. And then I had to go into a separate room outside the locker room to put on a mask and to put on a hairnet. And then I went into a hallway that led to the C and D cleaning rooms. When I had to go into another room before going to the C room. I had to change my hairnet and gloves. I had to put on a second set of gloves. And I also had to put on a set of coveralls and 2 sets of booties. Okay. That was the requirement for the C room. And I also had to spray my gloves with alcohol spray before I entered the room. So when you're talking about microbial contamination, you're talking about cleaning or preventing microbial contamination. It has to do with the stuff on the screen like HEPA filters and things like that, but also the flow of going into an area, going from like the dirty area, like in my street clothes, to the cleaner area with my scrubs, and to the cleanest area with the coveralls. Okay. So there is generally a system in place for cleaning and disinfecting room and equipment. So you can have daily cleanings, weekly cleanings, and monthly cleanings. A daily cleaning might just be going over the room with a certain type of spray. A weekly cleaning might be scrubbing the equipment and floors. A monthly cleaning would most likely be scrubbing the walls and floors and the equipment and using a respirator to do so, because you're working with cleaning materials that are so harsh that you have to have a respirator if you're going to be exposed to it more than a certain time limit. PowerPoint seems to have frozen. Alright, equipment regulations. Validation talks about how a company documents evidence to ensure that a process consistently meets predetermined specifications. Qualification is the testing of equipment, processes, and facilities to prove that they work correctly and lead to expected results. So validation talks about showing evidence that a process is consistent. Qualification talks about a process or equipment leading to expected results. Equipment must be validated and qualified before being used in biopharmaceutical production. Calibrations is the process of ensuring the accuracy of an instrument performing a measurement by comparing the measurement to a reference standard. For example, you might have a scale that you'll have a set of weights, and you'll take that scale and then you'll put the weights on it, and it has to read that weight exactly. And if it doesn't, you'll need to adjust the scale until it does read that exact weight. If you look like in labs, you'll have scales and it'll have like a little bar on the back that'll show that it's balanced and calibrated. For lab scales, larger scales have different methods. In order to do calibrations, you'll have a standard operating procedure to follow. So, Preventative maintenance prevents problems. Corrective maintenance corrects problems. Pretty straightforward. Ensuring quality when you're bringing in materials into the biomanufacturing facility. You need to make sure that you specify what material you need. Qualify vendors, meaning getting vendors that have quality products. Place the order with that vendor, receive the material. You're always going to have to sample and test the material no matter how many times you've gotten that same type of material from a vendor before. You always have to isolate it and test it before you can use it in production. Right. Any type of containers that materials are going to have to be labeled with the material name, part number, batch number, expiration date, material or initials of the person who filled the drum and wrote the label, and the date. So, when we talk about traceability, I like to think about packages because I order way too much stuff on Amazon. And I'm really glad that I don't live in like 1885 where if I order a package from somewhere and it goes missing, I have no clue where it went or where the issue was. Now, when you order something from Amazon now, you can track it, right? And that's the idea behind traceability. Forwards traceability means that you are tracing the material from the time of receipt until it goes into a batch. Backwards traceability means you can go from that batch all the way up back until the material was received. Okay? And the idea behind that traceability is if there's a quality problem with a batch, you can have forward and backwards traceability in order to figure out where the point was that the issue came up. So that way the problem can be solved quicker. So here we are in section 5. So we're going back to the 21 CFR. According to the 21 CFR, there shall be written procedures for production and process control designed to assure that the products have the identity, strength, quality, and purity that they purport are represented to possess. Deviations refer to events that do not conform to the instructions of approved procedures or good manufacturing practices. Some major GMPs include the idea that batches must be formulated to meet the full label claim for the active ingredient. So labels, the label into which the material is dispensed has to have the material part number, the batch number, the weight, and the product lot it was weighed out for. Line clearance just means that you're cleaning everything in between batches to prevent cross-contamination. Equipment verification ensures that equipment is released for use. That means it's been calibrated, it's been cleaned, it's good to go. Area cleaning I had talked about a little while ago. An example with the daily, weekly, monthly cleanings. Prepares production rooms and other areas before being used. This reduces the room's microbial load and risk of environmental contamination. Documentation checks help operators to ensure that they are using current versions of all SOPs. SOPs, you can have different versions of it. They can be rewritten, and sometimes like even the procedure doesn't change, but it's just written to make things more clear. And so you always want to make sure you have the latest version of your SOP. Yield and reconciliation calculations. And so a yield, we're talking about how much product is made from a batch. You can be asked to calculate theoretical and actual yield. A low yield is generally indicative of a lot of rejects in products. A high yield, usually you don't have as many rejects as you normally do. If you're having a particularly low yield or particularly high yield, there needs to be an investigation. If you're not sure what constitutes a low or high yield, ask. Ask. Always ask. Ask. If you work at a pharmaceutical company and they don't encourage you to ask every question you can possibly ask, you need to find a different place to work. Because that is part of safety culture. If you're not sure, ask. And if you're not in a place where you're comfortable asking because it's toxic or people are going to assume that you're, that you don't know everything you should know, you need to work at a different pharmaceutical company. Period. You should always be able to ask questions. Reconciliation is the process of accounting for all starting materials after processing. So parameter limits, when you're working with anything, particularly batches, let's say you're working in solution prep and you make a batch of solution, you can take a sample of that, and that sample has to meet certain parameters at a certain temperature, pressure, what have you. If the parameter has an impact on the process, then it has to have a defined acceptable range, as explained on the standard operating procedure. So one of the most important process parameters is time, and here are some examples of limits that are applied to different processes. So many processes require that a sample be removed at certain points to be analyzed. You should get instructions about how to collect samples and what the parameters are for those samples and how to properly label and submit those samples. Equipment qualification is the process of establishing equipment consistency. Process validation is the qualification of a manufacturing process. So you have primary packaging, which separates the material from the environment. Secondary packaging goes around the primary packaging. Container closure integrity is evaluating the barrier formed by the primary packaging to separate it from the environment. Each container should be inspected for defects. The inspection process is designed to identify damaged product with particles, or any number of other. So we've talked a lot about labels so far. We're going to keep talking about it. Labels are tightly controlled. Labels are directly regulated by the FDA, and each product has to have approved labeling, and variance from that approved label is a CGMP violation. So in this case, we're talking about labels that go onto the outside of the drug itself. Warehousing distribution. Product storage conditions are defined during product development. Storage usually has like time and temperature parameters, for example. By the time a product is finished being manufactured, the storage conditions are well established. Distribution is the transport of the product from the factory through distributor warehouses to clinics. So there are two goals of distribution: to protect quality and to ensure product traceability. You have active temperature control, which uses heating and cooling to ensure that the interior of a shipping environment remains at the desired temperature. Passive control uses insulation to reduce the rate of temperature change inside a shipping container. Both of those can be used to keep things like vaccines that need to stay cold within the temperature parameter throughout its entire time, from the time it leaves the manufacturer to the time it gets to the clinic. Sometimes distributors return product because it was defective or they ordered too much. A recall is a voluntary action that takes place because manufacturers and distributors carried out their responsibility to protect the public health. So a recall can apply to many different industries. I have a safety recall on my car that I have been lazy and not taken in to get fixed, even though it would be free. So recalls happen in a lot of industries, not just the pharmaceutical industry. The FDA doesn't force anybody to do a recall, but they will highly suggest it. Wink, wink, nudge, nudge. And if it is not done voluntarily, then the FDA will be up their tail, so to speak, and watching every move they make. So a recall is just easier than having the FDA breathing down your wet neck and watching every move you make. Laboratory controls are important. Even though laboratories are separate from the production area, laboratory is important in terms of like testing raw materials, testing samples. Water throughout the company is also tested to make sure, there's not contaminants or anything in the water. Finished products are tested and stability samples are tested to compare and talk about the shelf life specifications. Specification accuracy. So whatever specifications you're trying to meet must be scientifically sound. They must be approved by quality. Sampling plans must also be scientifically sound, and specifications should identify the test method against which the material is being tested. Testing shall be done by approved procedures and methods, so follow the SOP. Samples need to be adequately identified. Again, your SOP should tell you how to do that. Laboratory instruments should be calibrated and qualified. Test methods are qualified to be repeatable, to be accurate, and to give the same results without different analysts performing the test method. And comparing those results with specifications means that the raw data supports the result and is fully traceable. The decision is reviewed and approved by a second person, often in quality. Electronic systems and records. These are examples of different systems that different pharmaceutical companies will use. It just depends on where you work and how they choose to document. And here's your opportunity to review and your assignments